Appreciate you all being here this morning. We got two sections of this. Been an interest in going first in this virus that we have, the cotton leafroll dwarf virus. By the way, my name is Bob Kemerait. I'm with the University of Georgia. This is Dr. Sudeep Bag. He is with the University of Georgia as well. He is a cotton virus researcher, or virologist in general. I'm the Extension Plant Pathologist. First 10 minutes of this presentation, we'll be talking about the new cotton leafroll dwarf virus. The presence, and I'll mention a little bit in my talk, but the presence has been misconstrued in the past as, is it blue disease? It is not blue disease. There's been some questions. Is it related to bronze wilt of past? We really don't know, but some symptoms are similar. But I want to ask Dr. Bag to talk first with his presentation on this. Anybody who's not interested in the rest of it, which is more of the nematodes and the foliar disease management, you may find someplace else. All right? Dr. Bag, thank you. Now, you might have to describe it from the yes.
Good morning. I will be talking about the new virus that is making sounds here for last 2 years. So this is what we are calling it as cotton leafroll dwarf disease. That's the virus that is causing here. And let's go. So this is just my team. I would like to acknowledge everyone present here as well as the team member for the support. So now it's the winter season is going on, and how do we react to the cold? If we have the virus, cold virus, early in the season when we are younger, we are reacting it differentially, having a different type of symptom, but the common sense is the running nose. As the season grows and we get younger, the symptom changes, but it remains the same, the virus. You may have seen this type of pictures. First one is in tobacco, then in peanut, then in tomato, and then onion. If we see any of these symptoms, we know that this is spotted wilt or similar type of virus, and we don't have to do any further diagnosis. But we are not in that stage for this new cotton virus. This virus is showing different type of symptoms. As Dr. Kemerait mentioned, it is very much similar to bronze wilt, but we are still learning about it. Here you see the symptoms are like drooping, the leaves are going downwards. It is what we are seeing, and we are able to find the virus, detect the virus in this type of symptomatic plant. The other type of symptoms that we are seeing here is what we are calling as a crashing symptom, where the plant is crashing from day 1 to day 10. In this 10 days, the plant is crashing. And if you see the date, it is like 7/30, July 30. But those plants are crashing, but they are not dying. Those plants, under some change in the environment, it may be better condition, these plants are reviving. And they are again not showing any more further symptom, but these plants are not retaining any bole. These plants, all these plants that are symptomatic earlier, they are having zero yield. And we are able to detect the virus in both when they are showing symptom as well as when they are not showing symptom. Similar type of symptoms, we have seen it in Bleckley County. Here we have seen it much more severe, that the retention of the square, they start falling, less and less retention. All the leaves are falling down. And these plants, again, they revive. Another type of symptoms that we are seeing here in this year is in Bleckley County, where we are seeing this short internode. So this is all what we are seeing is late in the season, in August. The redness of the leaves or drooping that we are seeing in July. As we are progressing, the symptom is changing. So in this case, we are seeing short internodes, or we are seeing longer plants. In this case, we are seeing here different shorter internodes, or at the last one you can see, in that one it is much larger and there is no ball in any of those plants. We are able to detect in this type of symptomatic plants also. The other type of symptoms that we see is in Bleckley County, where we are seeing it like a bushy top. And in this case, also we are not seeing any ball retention or fruit yield. At the end of the season, there is around 10 to 15% of disease incidence in this field, and this type of symptom is seen only in Ben Hill County. So there is lot of different type of symptoms within Georgia, and they are with response to different geographical location or different environment present in each of the other counties. This is what we have seen in Tifton. In this particular plant, there is like 39 internode, not a single ball formation, not a single fruit. And if you see, it is in September 10 and by September 25 at the time of harvesting, there is no single ball in this particular field, in particular plant. What we are seeing it in the flower. So if you see this picture, in this one is the flower from a asymptomatic plant where we are not having any obvious symptom. So stamen is larger. Here you see there is a different, one is from asymptomatic and others from symptomatic. Finally the fruit, the one is the normal, whereas the other one is like a parrot shape, where we are seeing from the symptomatic and asymptomatic plant. What we see if we break it inside, from the fruit that is normal, we have around 8 to 9 seeds in each of the locks, whereas in other case we have only 2 to 3 seeds in each locks. So there is yield, and that is the impact due to this particular virus. Oftentimes we confuse ourselves with the symptoms that we may see due to the presence of aphid. So these symptoms what we are seeing here, these are not due to virus, but due to the presence of large number of aphids on this plant. And we are able to see that the virus is also there, but aphid is the responsible for this type of symptoms what we are observing. This is the what we can see as an impact. This is in our field trial where we have seen that this plant that is not showing any symptom, whereas the symptomatic plants, they remain dwarf, zero yield. This is how the plant will show at the end when it is infected very early in the season. Now at the last one, if you see, there are all orange flags. So it is like 50 feet from here till the end, and there are like 79 flags. So in 50 feet of distance, there is 79 plants not having any yield. So that is the impact is there. Are we able to detect in all the plants? So early in the season, if we are seeing we are having difference, asymptomatic plants, they are good. Symptomatic plants we are able to detect, but as the season progresses, most of the asymptomatic plants, virus is there, but we are able to detect, and but they are not showing any symptom. This is in Georgia, where we have, in which all counties where we have got the sample collected, where we are able to detect the virus. It doesn't mean that all the white spot virus is not there. It's just that we are not able to get the sample. It covers the entire cotton growing region in Georgia where we have this virus present. So how can we manage it? First thing we need to avoid. How we can avoid? Avoid the aphid, avoid the planting date, or if we have a cultivar that is resistance. We are yet to research in that one. We can eradicate the pathogen. How we can do that? Cultural practices. How we can do field sanitation. That is an important thing. And finally, the golden bullet is having resistance, plant host resistance, and some transgenic or those aspects we can develop the plant. In this particular one, when I say avoidance, we need to maintain the sanitation. We have seen lot of different weeds. This particular field is 1230. In December 30, we see there is a lot of henbeats, as well as the cotton plant is after harvest is there. And we see here there is lot of regrowth as well as weeds. So these regrowth that are present in the field, they are harboring the virus for the next season. So we tested around 30 different weeds, and we are able to detect in number of different weeds. Just detection of the virus in the weeds doesn't say that this is the suitable host or a vector for the vector or the virus, or it's a potential green bridge. We still need to study that before we can say which weeds are important, which weeds are not. But at this point, we need to take care of regrowth, we need to take care of hindweed, we need to take care of pigweed, where we are seeing more and more number of virus present.
Dr. Bank, just say clearly, so far you have found that hindweed and pigweed, the Palmer amaranth, and what else have been tested positive using PCR for the virus?
White clover, perennial peanut, but just as I said, just detection of the virus in those, I will not say that those we need to take care unless we do more studies on that of transmission, whether they are doing or not, whether the virus is replicating or not. Just finding that, but these are the where we are testing. If we are testing 9 out of 10, we are getting the presence. In other case, it is very less frequency. So we need to maintain that which is more potential than the other. So finally, I will stop here. This is a field that we have seen in September 18, very beautiful field in Bleckley County. And I just stop by and take 2 samples. The symptom you can see where you have half of the 50% of the plant is showing yield, whereas other half is nothing, and we are able to test the virus. This means the virus is present in this field, but it is not impacting. It not impacted this year. What is going to happen next year, we don't know at this point. Finally, this is the field we have seen, or similar field we can still see in Georgia, depending upon where we are driving. These are the potential host or the potential reservoir for our coming season. How can we avoid the minimum amount of inoculum or virus for our next season? We may have to take care of this one. Finally, I would like to thank my team and stop for any other questions.
While we're on this topic, and I was going to cover a little bit later, but no need. Questions for Sudeep on cotton leafroll dwarf virus. Again, it's not blue disease. It is a new virus related to one out of Brazil. It is aphid transmitted. Okay? Questions for Sudeep.
What was the percentage of asymptomatic plants that showed positive for the virus?
So if the sample is collected late in August, early in September, 95%.
Dr. Kemerait, is that with tomato spider wilt? Doesn't one in the 80s, around 80% that you can detect with TSWV?
I'm not sure of that. I think the take-home message on this is that if we look at fields across the state, we can say we know of 2 in 2019 where we feel there was significant yield loss associated with it that we can document. If you're to ask us how many percent of the fields were infected in some degree or other, I believe most of them were. If you're to ask us what we think of the yield loss from this virus in 2019 statewide, very, very small. The concern to us is based upon what he said. There were a large number of asymptomatic plants out there that if they were to ever trigger symptomatic, could be very, very serious. We don't know exactly how long we've had the virus. We don't know where it's going from here. That's why the effort's going into it. Certainly deserves effort. Okay? More questions for Sudeep.
By green bridge, do you mean cover crop?
Right now, I'm not talking about cover crop because the rye or the monocots that we are using as a cover crop, we have not seen it. Green bridge here, I mean to say the weeds that we are having right now in and around field and the stock remaining from the last season. That is, now we have a regrowth in those ones. If we do not have the freezing and if the stock is not dying, that stock is the potential reservoir for our next season.
And so in terms of green bridge, when he uses that term, he's referring to what could possibly carry the inoculum, the virus titer, from 2019 into 2020. And right now we recognize that that bridge, that what could carry the titer could be the regrowth on the cotton, or it could be winter weed hosts that are out there that are known to be test positive. At this point, as he mentioned, we don't know how effective they would be. The henbit, the Palmer amaranth, the white clover if it was grown here, the perennial peanut, we don't know how effective that would be as a green bridge other than the fact that he has confirmed that it is present.
You call it leafroll disease?
At this point, I'll let him
At
answer.
this point, it is cotton leaf roll dwarf virus.
Does it
And
roll the leaves? Because I've
As-
seen that and I laid it to some type of foliar application. I'm just,
so it depends, the symptom depends on number of different factors. One is the time of inoculation or when the plant is get infected, early in the season, late in the season, temperature, plant health, soil, all other factors. Early in the season we have seen that leaf is changing or drooling or downward curling. In many cases we have seen it upward. We have not seen this redness drooling anywhere in Colquitt County or in Ben Hill County. There we have seen totally different symptom. In Bleckley we have seen totally different symptom. So within this small radius we are seeing different type of symptoms. So it is the variety response will be there, time of inoculation, plant health, local temperature, and then whether it is dry land, whether it is irrigated, plant stress. Whether if it is in the stress and we have virus, that will be more severe. If the plant is healthy and we have virus, it may remain asymptomatic, virus may remain present there.
Mark, one of the frustrations is if I show you a peanut with tomato spotted wilt virus, we're both going to agree that's tomato spotted wilt virus. If you look at the symptoms that have tested positive for cotton leaf roll dwarf virus, they could also be confused with symptoms of fusarium wilt in the field. They could be confused with seedling disease. They could be confused with chemical injury. They could be confused with aphid feeding. They could be confused with a number of different factors. And so as he's mentioned, depending upon the conditions under which the plant is, the symptoms may vary. The bottom line for extension for University of Georgia right now is we continue to look at it. At this point in 2018 and 2019, losses were minimal across the state. Second thing, it's not clear how long this virus has actually been in Georgia cotton. We suspect that it's been here a lot longer than it was first detected in 2017 in Auburn, Alabama. The third thing is if we're given recommendations to growers on how to manage cotton leaf roll dwarf virus in 2020, managing aphids, and Philip Roberts in the back, managing aphids is not a key factor. You cannot kill aphids fast enough or completely enough to complete the transmission of the virus. The last thing is at this point, the only management strategy we have for cotton leaf roll dwarf virus is to avoid planting late. It seems to be more severe in later planted cotton. Okay? What I'm hoping is, how many of you all have heard of tomato spotted wilt virus before on peanuts? How many of you all have heard of peanut stripe virus? 1990s peanut stripe virus was a huge concern among breeders and amongst virologists. Peanut stripe virus is still there. It doesn't matter. Tomato spotted wilt virus does. We want this to be a peanut stripe virus, but not a tomato spotted wilt virus. But that's why we have our best people working on it in conjunction with our team, in conjunction with Auburn University, Mississippi. Tom Allen's with us on this too, Mississippi State, trying to figure out what the response is. Okay?
What about wild radish as a green bridge? Is it,
We have tested it, we have not yet detected
You have not.
The virus.
They've tested but have not detected the virus in wild radish.
A lot
So
of it
far.
out there, and a lot of people are spraying with 2,4-D now for wild radish control.
Okay. As of yet, though, he's not been able. He's tested wild radish. Question is, there's a lot of it out there. A lot of it's being sprayed. He's tested. We've not found it. Dr.
it's
Crawford, we
just
might
one-
find it later. I don't. At this point, we have
It's
not.
just one season, 1 year. What will happen next or this year? We are going in round this year also to collect weed, and we are keeping it testing this year also.
Other questions? Other questions? Cotton leaf roll dwarf virus. The most important thing you can walk away from this is the sky is not falling on it yet. We hope that it won't, but there's a considerable amount of effort. Auburn University has just gotten like a pile of money to look at it. We look forward to sharing that resources with them. So, any further questions on that? Thank you, Dr. Bagg, for that. Any further questions? Okay, I'm going to go in the 30 minutes I got left. I'm going to talk about things, problems that we've had before, problems that we continue to look for solutions, problems we have new solutions for. I do want to start off with this slide. And this slide is Dr. Bagg talked about green bridges. They've never been more important. If you look at the picture on the right-hand side, this was in January of 2017 in Mitchell County, Georgia. And at that point in time, Rick, the biggest problem we had with that when I talked about it was if we've got cotton surviving, if you've mowed it but it continues to survive, the biggest problem is it continues to feed nematodes. It continues to build nematode populations. The question comes into, is it worth destroying those stalks or not? We're getting the data. We're working with Stripling right now, collecting that kind of data. If you destroy those stalks, what happens at harvest? What happens to nematode populations? It's become even more critical. And again, I cannot put a monetary value on what's the value for the virus, the cotton leafroll dwarf virus, in destroying this or the impact of winter weeds. All I can say is they're not doing anything good. At best, they're not helping or hurting. At worst, they may be serving as a green bridge not only for nematodes but also for the virus. And that's why we continue to look at it. The most important information we're trying to gain now is what is the economic value. If you go out and you buy a $13,000, $14,000 piece of equipment to destroy those stalks when you harvest, what's the return to you? If you don't get a hard freeze or stays cold until in February, it's probably going to be more valuable than if you get a hard freeze at the end of November. Okay? We talked about the cotton leafroll dwarf virus. That is a sexy situation. That is new. It is one that people are worried about, and for rightful reason. But I want to focus your attention that what's in my wheelhouse in disease nematode management in Georgia, cotton leafroll dwarf virus, based upon its impact as far as what it has done so far, is very, very low down my list of things that you can do something about or that you need to do something about. That may change. We can't afford to sit back and wait. But nematodes remain the most important problem we have as far as my wheelhouse goes. Okay? And you know that. We've done this before. But if I was to list what I think as far as the problems growers should be aware of in the list of importance right now, based upon what happened in 2020, I would put nematodes at the very top. Whether it's root knot nematodes, whether it's Reniform nematodes, whether it's sting nematodes. And below that I put target spot. Not because target spot takes yield out of every grower's field every time, Scott, but because there's opportunity to manage it. There's opportunity to spend money, there's an opportunity to decide, and everything below that makes a difference. These are what's important. Cotton leafroll dwarf virus is on there, but so far management options are limited and yield loss is limited so far. Okay? I'm going to drive through this in the interest of time. Seedling diseases, all I want you to remember, nothing new about that, but just remember when we talk about you get one chance. You get one chance to make the right decisions. You get one chance to fight seedling disease. If you get two chances, it's because you have to replant. You get one chance to fight nematodes. When we're talking about seedling disease in 2020, your decision on top of what we put as far as comes on the seed, whether you use an in-furrow fungicide, whether you use an extra seed treatment, Syngenta, Wilson's got your new seed treatment on top coming out, whether you use in-furrow, whether you use initial seed treatments, has to do with what your field history, has to do with your environment. Is it cool and wet at the time of planting? What's your planting date? The important thing here is not that you haven't heard this, it's to recognize you get one chance, and the opportunities out there are seed treatments on top of it and new fungicides that can be put into effect. If you're using Velum Total for nematode control, Velum Total will also offer some benefit on that. Okay? Let's talk foliar diseases. And again, target spot was second on my list, not necessarily because the yield loss justifies that, but because there is the opportunity to do something about it. And that opportunity continues to expand into 2020, what fungicides are available. Also, what's new is this Areolate mildew. Back in 2005, the first time I saw Areolate mildew, it was more of a harvest aid. It was something that came in late. As of 2017, 2018, and 2019, if you're growing cotton or you're talking with cotton farmers, Areolate mildew takes an increased importance because you have the opportunity to manage it. And the question comes, if you don't manage it, does it matter?
And the bottom line on that is based on work out of Colquitt County, based on work out of Brooks County. If you have the opportunity to manage areolate mildew and you miss that opportunity, it can cost you 200 pounds of lint if you need it. We'll talk more about it. Foliar diseases are more important than they have been in the past. Target spot, again, we're familiar with target spot. The most important thing to recognize about target spot is in a dry year, like last year, target spot takes on less importance. In a wetter year, as earlier the target spot comes in, the more severe it will be. From the upper left-hand picture to the lower right-hand picture can take a matter of 2 weeks when you have good growth cotton, high fertility, high humidity, rainfall, and irrigation. And if you miss the opportunity, to manage this disease, then there's little you can do to catch up. If you don't need to spray for this disease, if it's hot, if it's dry, if you don't have good growth, if you can walk through a cotton canopy at 1 o'clock in the or 10 o'clock in the morning, walk through a cotton field at 10 o'clock in the morning in khaki pants like this, don't come back wet and green, don't worry about target spot. If it's 12 o'clock noon and you walk through a cotton field and your pants come back and they got moisture on them, those green streaks, you know that's where target spot will reside. Most important thing, what can we do about it? Well, the first thing we can do about it is ignore it and say it doesn't matter, which is what a lot of growers have done, especially in the eastern part of the state. You can ignore it and say it doesn't matter, okay? The second thing you can do, and if you do ignore it and don't do anything, you may save the money from having it sprayed for it. But if you do pay attention to it, and if you do treat for it, so far in Georgia we've seen protecting up to 200 pounds of lint. In Auburn they're reporting up to 400 pounds of lint. If 200 pounds of lint at today's cotton prices are not valuable to you, then we can skip this and go on. Don't even listen to it, okay? But if 200 pounds of lint is important, we need to talk about it. These are the fungicides, Scott, that we've come so far since 2005, which is now labeled on it. New for 2020 will be Miravis Top, okay? For target spot, the most difficult thing in controlling target spot is getting the material to the target. It's more of a problem when you have heavy growth, heavy dense foliage. Timing is everything. Out of all these fungicides, Preaxor has been the standout. In fact, all states agree that Preaxor has been the best as far as that. The others will help control it. Preaxor has been most effective. As of 2019-2020, Miravis Top from Syngenta is looking on par with Preaxor as far as controlling target spot. Okay? You have the opportunity right now. By the first week of bloom, we should be considering whether we need to spray or not. By the third week is a critical date. By the sixth week, it probably is either too late or doesn't matter anymore. Okay? Are we wasting your money? Are we wasting your money? This is data from several years, 2016 to 2018, looking at my trials for target spot. Orange is where Preaxor was sprayed once at the third week of bloom. Yellow is where we sprayed at the third and the fifth week of bloom. Okay? The number on top is the amount of lint on top of the untreated. So if you see 59.6, we got 59.6 more pounds of lint where we treated from where we didn't. If you see a negative 104, that's where we actually lost lint. Okay? To set this up, this bar, at the 65-cent cotton I was looking at, that's the amount of cotton you would need to make with one application to pay for target spot application. All the orange are above that. The second bar, that's the amount of cotton you would need to make at 65-cent cotton to pay for 2 applications plus the cost of application, and on top of that is the profit that came. 2 times we lost. That was a dry year where we didn't have target spot. It's a year where Michael came through and it didn't matter. Target spot in these situations, these were done in Attapulgis and in Stripling Irrigation Park, it did matter. Okay? Any questions on that?
Dr. Kemerait.
Yes, sir.
You may be about to cover.
No.
My question was, in dealing with areolate mildew, should we continue the same fungicide regimen as
Excellent
we do
question.
with target spot?
Excellent question. And that's you led me right into it, Brandon.
Okay.
Areolate mildew, until 2017, I looked upon this as, well, that's curious. It comes in East Georgia. It's a defoliant, it's a harvest aid. As of 2017, somehow it started coming in more aggressively. It came in earlier and it didn't stay home in southeast Georgia. We were finding it in southwest Georgia. They were finding it as far as Arkansas, Mississippi, even Texas. I cannot explain why that happened, okay? But I can tell you the question is, does it matter? First off, to tell you, with areolate mildew, it can be very aggressive. It can be very aggressive. The second thing, it is much easier to control than target spot is. Much more exposed, much easier to control. Much easier to the fact that we can get very good control of areolate mildew with even azoxystrobin. Okay? And there's a big price difference between Priaxor and Mirabis top. There's a big price difference. Okay? So azoxystrobin can be used effectively to manage it. Doesn't matter. Okay? Here's trials out of Colquitt County, 2018. On the right, on the left in blue is Quadris or azoxystrobin. That's the amount of Lint. The untreated was 719. A single application, well-timed, of azoxystrobin went from 719 to 863, 150 pounds of Lint. On the right, similar trial, Colquitt County, 688 pounds of Lint to 821 pounds of Lint with Priaxor. Take-home message there is that the azoxystrobin is giving us similar control as we would with Priaxor. So the bottom line of your question is we do not, if it's target spot or threat of target spot, Priaxor or the Mirabis top is my choice. Can you get by? Here's one from Brooks County. Same disease, 2 applications, 2018. 1196 Lint, 1286 with a single application, 2 applications, 1361. So azoxystrobin is a less expensive way to do it, but azoxystrobin is notorious for resistance developing. My biggest concern on here is not what it costs the growers to use azoxystrobin to control areolate mildew in the short term, it's what's the risk of using a single mode of action in the long term. And that's why products like Priaxor, Mirabis top, and those which are combined will last longer. Okay? But in the meantime, we can't get control. If we have any threat target spot, I would not use just azoxystrobin. If it's only areolate mildew, a grower needs to ask themselves, what's the value of resistance management? I think it's critical. When will we spray for this? If you're within 4 weeks of defoliating your crop anyway, you're within 4 weeks, I would not spray for it. I would not. Because you're already going to have made most of your fruit, all it's going to do is help you defoliate. But if you're 4 more weeks away from defoliating, and you find this, and you haven't already lost 20% of your leaves, you found it, but it was too late, I would spray for it. I would spray for it. Yes, Mark?
So you're saying these applications are not preventive applications? This is if you have a history in this field or you already find
Great
the
question.
mildew or the target spot or something?
What's the first principle of using fungicides effectively is get there firstest with the mostest, right? Get there first. You know where that came from, don't you? Get there first with the most, okay? In this situation on peanut leaf spot, you know peanut leaf spot's coming. You want to be out there preemptively. On areolate mildew, you do not know if it's going to be there or not. You can spray preventatively. There's nothing wrong with doing that, but the issue for me is we don't know when it's going to come and we don't know where it's going to be. And for that reason, I suggest if you're watching, if you're scouting, if you're looking for it, I would not spray until I either know I have it or it's in the general area. Okay? I would not spray if you get out there and find I missed it. I've lost 30% of my leaves. I would not spray for it. Okay? But if you know that it's coming, or you know neighbors have it, you know you have it, and conditions are favorable for spread, then I would spray treat. The data supports that.
Same thing with the target spot.
With a caveat. The target spot, so his question is the same thing with the target spot. I'm saying with areolate mildew, my recommendation is don't start spraying for it until you know that it's imminent. Either you've got it, in a little bit you're finding it, or your neighbor has it. Okay? Target spot, that's a good way to do it. Don't start spraying until you scout for it, or until you see it. With the exception, if you're a high input grower in southwest Georgia who's battled this disease many times, you've got a good crop coming, you've got the foliage, you've got the yield potential, a preventative spray at the 3rd week of bloom is not a mistake. Okay? Not a mistake. I still would like you to be out there looking. Now, if you get out there, if you're that grower, and you get out there at 3rd week of bloom, Mark, and you see there's nothing out there, I wouldn't spray. But if you say I got a lot of acres to cover, I've got a healthy enough respect for this disease and what a good fungicide will do that I would treat for it. But if you're looking and you're not finding it, that's a different story. Other questions on that? We've gone from not recommending any spray for foliar diseases, now there's opportunity. Okay?
What was the timing on the sequential treatment, and was the disease present at the time of the first application?
Great question. I believe, and I have to go back and check with them, I got the notes, but it was about the 3rd week of bloom, and there was the first one, there was nothing there. They were spraying. It's a good question. On those trials in Colquitt County and Brooks County, those trials were set up just to see what would happen. The first application went out before any disease was present. Okay? That's one of those situations where we could have sprayed and nothing came in. In this situation, we sprayed, they sprayed, and it did come in.
So you were spraying for Cornespra, and the second shot showed a benefit.
They were spraying, in this case, they were spraying in anticipation of areolate mildew.
At the third week of bloom?
At the third week of bloom. They were anticipating. That was, it was a lucky that we got that data. Okay, we did not know what was going to happen. They said, well, when can we go out there and spray just in case it comes in?
One observation I would add, where we spray a fifth week of bloom treatment for Cornespra target spot, we don't see areolate mildew behind it. I won't say that happens every time, but as a rule, we don't see it behind it.
Okay, y'all hear that? If you're spraying preventatively, or you're spraying in anticipation, or you're spraying as part of a target spot program, you don't see areolate mildew come in after it, and I would agree with that. I would say that the fungicides you're spraying for target spot at the fifth week of bloom are strong enough that they're going to take areolate mildew out of the equation. So whether it was coming in or not, I can't tell you, but if you're spraying for target spot anyway, you are taking areolate mildew out of the equation.
Almost without exception, the second and third week of spray was sprayed prior to that.
Good. Absolutely, I agree with that. Other questions? Other questions? All right, we talked about this. If you missed it, it's not blue disease, it's cotton leaf roll dwarf virus. Dr. Bagg has gone over the symptomology. The biggest problem in symptomology on the cotton leaf roll dwarf virus is it can look like a lot of other things. Cotton leaf roll dwarf virus is confirmed across the cotton belt from, I think they've got it in, they've certainly got it in Louisiana, they got it in Mississippi, got it in Florida, got it in Alabama, South Carolina, I believe has it. I think Tennessee has it as well. How long has it been there? We don't know. At this point, managing aphids is not the answer. At this time, planting later is not a good thing, but we don't know what the implication is going to be. But anyway, we've talked a lot about that already. Okay? Last thing I want to talk about in the next 10 minutes. Okay? And that is something that really does matter. Really does matter. And you would think as long as root knot nematodes, and nematodes have been a problem on cotton, we could talk about something else, but the problem is they continue to become more and more important. I always get the question, is it too late to pull nematode samples? Well, the general answer at this point in the season is it is. Okay? You pull those when it's warmer soils out there. But if you had plants where you had poor growth or where you had any kind of root knot galls like this, you don't need a soil sample to tell you got a problem. I want to remind everybody, not that you need to be reminded, this is out of Washington County a few years ago. It looked like nematodes, it acted like nematodes, but there were no galls on the roots. And so the grower was confused. It couldn't be nematodes. I want to remind you that only the root knot nematode produces the galls. The sting nematode, which is devastating down here, the Reniform nematode, which is devastating elsewhere, do not present with galls. You can only tell those really from taking soil samples. So what does it matter? What does it matter? A nematode's a nematode, a bad one's a bad one. Who cares if it's sting or it's root knot or it's Reniform? It matters because when we talk about managing nematodes, we're getting closer and closer and improving the opportunity for management of southern root knot nematode. At this point, not Reniform yet, not sting nematode. We're getting closer and closer with our opportunity to use resistant varieties for southern root knot nematode. If you've got a sting nematode problem, we do not have resistance. If you've got a Reniform nematode resistant variety, you're going to have to wait till next year when Phytogen brings out a Reniform resistant variety. At this point in time, we have varieties that are resistant to southern root knot nematode. Okay? You get one chance to plant them. You get one chance to plant the root knot nematode resistant variety. Okay? You have to make a decision. In this situation for 2020, what's new? Phytogen 400 has a single gene for resistance. Phytogen 500 has 2 genes for resistance. Okay? The root knot nematode, these complement what we already have up there. All of the Phytogen varieties are also resistant to bacterial blight. Resistance to root knot nematode and resistance to bacterial blight. Stoneville 5600, Delta Pine 1747 are bacterial blight susceptible, but they have the root knot nematode resistance. You need to make a decision. Do you want to use a root knot nematode resistant variety? If it's a Reniform field, you have to wait till next year. If it's a sting nematode field, we don't have resistance. But you get one chance to decide on the planting of it. Show this slide. Okay? All I want to point out from this slide is what keeps more growers from planting root knot nematode resistant varieties. It's hard to look at a top yielder and then look at what a root knot nematode resistant variety has traditionally done outside of a nematode field and to feel good about planting that. The point I want to make is these are variety trials, county variety trials last year. Phytogen 400, single gene for resistance, new variety for 2020, was one of the top performers in terms of yield. That doesn't mean you have to plant it. The take-home message here is that the breeders are coming closer and closer to giving you the yield potential you want and the resistance to the root knot nematode in the same variety. Okay? The biggest reason more growers have not planted these varieties, despite what I've talked about resistance, is the concern over yield potential. Okay? What do you get? What do you get when you plant a 480, a 1747, a 5600 versus a 1646? Okay? This is one trial. It doesn't always look like this. But this is a root knot nematode field. This was out of Packer Park in Colquitt County. This is the amount of nematodes left at the end of the season. The soil counts at the end of the season. That's 1646. That's 2.5 times our threshold. The use of Temic lowered a little bit, but if you went with 5600, 1747, or the 480, you had almost no nematode buildup. Again, that's one of the advantages of using it. And again, this is one trial. It does not always work this way. But if you look, here's the yield potential in that field with 1646. When you added AgLogic to it, you increased your yield by 100 pounds. Okay? When you look, 100 pounds of seed cotton, when you look at the root knot nematode resistant varieties that had no nematicide with them, you see the yield benefit. Does that happen every time? You know as well as I do, it doesn't. But that's what the potential is. The opportunity to plant the variety without using a nematicide.
Dr. Kemerait,
Yes, sir.
3.5 pounds of AgLogic or 5?
This was 5 pounds. 5 pounds of AgLogic. Good question. 3.5 pounds, Brandon, is our thrips rate. 5 pounds is what we're putting out there for nematodes. Great question. Now I can show you other trials where 1646 with either Velamitodal or AgLogic is going to out-yield those varieties. There's lots of them out there. Okay? But this is an example of a situation where our nematode counts were such that AgLogic at 5 pounds was not enough to bring the 1646 up to what the resistant varieties did.
Let me ask you another question.
Sure.
You know, we got growers that are teeter-totting, using Infuro, Vellum Total and wanting to switch to AgLogic. I mean, is it worth the cost to, 16 ounces of Vellum versus 5 pounds of AgLogic?
Great question.
Just to switch the whole operation.
And I'll come back to your question here in a second. If you are going to plant in a root knot nematode environment, you're going to plant anything other than a root knot nematode resistant variety. You may do it for a lot of reasons. The variety looks better, you like it better, you like the yield potential, whatever reason, you owe it to yourself, Brandon, to go out there and use a nematicide. And the types of nematicides we've got are increasing, okay? And your question is exactly why we continue to talk about it. Because right now, Telone 2 remains the most effective, most important nematicide we have on cotton and on peanuts. And for the first time in my career, it's not in short supply this year. It's not in short supply. Very, very effective. But for most growers who need a nematicide but are not going to put it out because of whatever reason, that leaves them, we'll talk seed treatments in a minute, is whether they're going to use AgLogic or Vellum Total. My experience, AgLogic, for your information, is a granular insecticide, nematicide. You have to be able to put out a granular. Vellum Total comes as a liquid. It can be put out in furrow. It also helps with seedling disease. The question you ask is a question I get all the time. Why would I go with one versus the other? Strictly from a thrips standpoint, AgLogic is going to be better on thrips than Vellum Total, which relies upon imidacloprid. But this isn't a thrips talk, okay? I would say, from my experience with our data, that 5 pounds of AgLogic is approximately equal if you're putting out 16 to 18 ounces of Vellum Total. If you're going to go with 16 to 18 ounces of Vellum Total versus 5 pounds of AgLogic, the results on the nematodes are basically the same. Better thrips control, perhaps, but the nematodes are about the same. Where does the AgLogic shine? If you needed Telone but you weren't going to put it out, 18 ounces of Vellum Total is not enough. 5 pounds of AgLogic is not enough, but you can bump that AgLogic rate up to 7 pounds. And 7 pounds of AgLogic will be better against a tough nematode situation than would have been the 16 to 18 ounces of Vellum Total. Okay? You got a cost. 7 pounds of Ag Logic is about $42 there and about. 18 ounces of vellum total is probably about $36. Okay, there is a price differential. You have to come up with that decision on what you want to do. Okay? Any questions on that? Mark, your question.
I think you're about to get to it with the seed treatment. We're
Okay,
seeing a
great.
trend, maybe some areas away from the seed treatment for cost. You know,
Okay.
some guys say, is it worth it?
Let's talk about seed treatments. Seed treatments bring what Telone can only dream of. Convenience. Seed treatments bring what Ag Logic and Vellum Total can only dream of, and that's convenience. You put it on a seed, you plant it. A lot of work has been done, Wilson, looking at Avicta. They were the first ones out there we were looking at. And we still got Eris, but it's more on thrips now. We have Copio Prime with BASF. We have Avicta. We have BioST. Forget Nemastrike. It won't be available this year. And it doesn't break my heart. Okay? But the ones you'll have an opportunity for will be Copio, which is the same fluopyram that is in Vellum Total. You got Avicta, which has abamectin, no stranger to that, and BioST. It's not a biological, but it's a derivative of a fermentation process. What I would say is in the right trial where we have low nematode populations, all of these can look good. Most experience with Avicta, Copio we got a lot of experience with too, they can look good. But what I would say this is my experience again, if you know you have a nematode problem, if you're going out there and you recognize you need to do something, these are probably not going to be what you need. In the sense of, not that they won't help, but it's more of a Band-Aid. If you know you have a nematode problem, if you've done your root counts, these are a stopgap perhaps. Vellum or Ag Logic is going to be better. Telone can be better than that. You do have the opportunity now with Return XL from AMVAC, you do have a return for a Vidate as a post-plant application. 17 ounces once, or split it into half that twice. You can complement these with that. But the results on southern root knot nematode have been mixed at best. Okay? Bottom line, what are your opportunities? You've got a nematode problem. If it's a root knot nematode problem, you need to make a decision. Are you going to use a root knot nematode resistant variety? If you are not going to use a root knot nematode resistant variety, like the 400, like the 500, 1747, 5600, you need to choose a nematicide to use. And the choice of nematicide comes down to what you can stomach as far as application and what you need is efficacy. Nothing better than Telone. Jared, it's the first time I've ever not heard you tell me, don't oversell Telone. We don't have enough. You got enough this year, I think. Okay? But if you're not going to use Telone, how does Vellum or Ag Logic fit in? The convenience of Copio, of Avicta, of BioST are well known. I don't have to tell you how convenient they are. The question mark is not are they convenient, it is are they what you need. Okay? I'm at the end of my time. I want to thank you all for being here. I want to remind, as you leave, I want you to remember one thing. Cotton leafroll dwarf virus is out there. If you have concerns, if you have questions, if you think you might see it in your field, we have the opportunity to look at it. Dr. Sudeep Bag is one of the leading specialists, one of the leading experts in the country on that virus. I'll tell you this, we don't need to check every single plant to see if it's there, but it does help us if we know you're finding it. Second thing is you get one chance to do things right. Are you going to use extra in-furrow treatment? Are you going to use extra seed treatment? Are you going to fight nematodes? Because if you don't do that, when you close the furrow, you spend the rest of the season wishing you'd done something different. And I'll say the last thing is this for foliar diseases, whether it's target spot or areolate mildew. You can believe they matter, you can believe they don't matter, but I think it's in the best interest of every cotton producer in the state of Georgia is to make a conscious decision. I'm either going to treat if I need to or I'm not going to worry about it. And once you decide I'm going to treat if I need to, you need to think about when you would do it and what product you would use. Okay? Any last questions from me? Okay? All right. I appreciate it all very much. Thank you.